Pseudomonas syringae is a rod-shaped, gram-negative bacterium with polar flagella. As a plant pathogen, it can infect a wide range of species, and exists as over 50 different pathovars, all of which are available to researchers from international culture collections such as the NCPPB, ICMP, and others. It is unclear whether these pathovars represent a single species. P. syringae is a member of the Pseudomonas genus, and based on 16S rRNA analysis, it has been placed in the P. syringae group. It is named after the lilac tree, from which it was first isolated. P. syringae tests negative for arginine dehydrolase and oxidase activity, and forms the polymer levonon-sucrose nutrient agar. Many, but not all, strains secrete the lipid-epsilon-peptide plant toxin syringamycin, and it owes its yellow fluorescent appearance when cultured in vitro on King's P medium to production of the siderophore-pyoverdin. P. syringae also produces INA proteins which cause water to freeze at fairly high temperatures, resulting in injury to plants. Since the 1970s, P. syringae has been implicated as an atmospheric biological ice nucleator, with airborne bacteria serving as cloud condensation nuclei. Recent evidence has suggested the species plays a larger role than previously thought in producing rain and snow. They have also been found in the cores of hailstones, aiding in bioprecipitation. These INA proteins are also used in making artificial snow. P. syringae pathogenesis is dependent on effector proteins secreted into the plant cell by the bacterial type III secretion system. Nearly 60 different type III effector families encoded by HOP genes have been identified in P. syringae. Type III effectors contribute to pathogenesis chiefly through their role in suppressing plant defense. Owing to early availability of the genome sequence for 3 P. syringae strains and the ability of selected strains to cause disease on well-characterized host plants, including Arabidopsis thaliana, Nicotiana benthamiana, and tomato, P. syringae has come to represent an important model system for experimental characterization of the molecular dynamics of plant-pathogen interactions. Epidemiology: disease by P. syringae tends to be favored by wet, cool conditions. Optimum temperatures for disease tend to be around 12–25 degrees Celsius, although this can vary according to the pathovar involved. The bacteria tend to be seed-borne, and are dispersed between plants by rain splash. Although it is a plant pathogen, it can also live as a saprotroph in the philosphere when conditions are not favorable for disease. Some saprotrophic strains of P. syringae have been used as biocontrol agents against pastharvist rots. Mechanisms of pathogenicity: the mechanisms of Pseudomonas syringae pathogenicity can be separated into several categories: ability to invade a plant, ability to overcome host resistance, biofilm formation and production of proteins with ice nucleating properties. Ability to invade plants equals, planktonic Pseudomonas syringae are able to enter plants by using their flagella and pili to swim towards a target host. They enter the plant via wounds of natural opening sites, as they are not able to breach the plant cell wall. The role of taxis in P. syringae has not been well studied. However, it is thought that the bacteria use chemical signals released by the plant to find their host and cause infection. Overcoming host resistance equals, P. syringae isolates carry a range of virulence factors called type III secretion system effector proteins. These proteins primarily function to cause disease symptoms and manipulate the host's immune response to facilitate infection. The major family of T3SS effectors in P. syringae is the HRP gene cluster, coding for the HRP secretion apparatus. The pathogens also produce phytotoxins which injure the plant and can suppress the host immune system. One such phytotoxin is coronitine, found in pathovars phtho and PGL. Biofilm formation equals, P. syringae produce polysaccharides which allow them to adhere to the surface of plant cells. They also release quorum-sensing molecules, which allow them to sense the presence of other bacterial cells nearby. If these molecules pass a threshold level, the bacteria will change their pattern of gene expression to form a biofilm and begin expression virulence-related genes. the bacteria secrete highly viscous compounds such as polysaccharides and DNA to create a protective environment to grow in. Ice nucleating properties equals P. syringae more than any mineral or other organism is responsible for the surface frost damage in plants exposed to the environment. For plants without antifreeze proteins, frost damage usually occurs between -4 and -12 degrees Celsius as the water in plant tissue can remain in a supercooled liquid state. P. syringae can cause water to freeze at temperatures as high as 1.8 degrees Celsius, but strains causing ice nucleation at lower temperatures are more common. The freezing causes injuries in the epithelia and makes the nutrients in the underlying plant tissues available to the bacteria. P. syringae has INA genes that make INA proteins which translocate to the outer bacterial membrane on the surface of the bacteria, where the INA proteins act as nuclei for ice formation. Artificial strains of P. syringae known as ice bacteria have been created to reduce frost damage. P. syringae has been found in the center of hailstones, suggesting the bacterium may play a role in Earth's hydrological cycle. Pathovars. Following ribotypical analysis, incorporation of several pathovars of P. syringae into other species was proposed. According to this schema, the remaining pathovars are Pseudomonas syringae PV Aceris attacks maple acer species. Pseudomonas syringae PV actinidiae attacks kiwifruit actinidia deliciosa. Pseudomonas syringae PV aesculi attacks horse chestnut Aesculus hyocastanum, causing bleeding canker of horse chestnut. Pseudomonas syringae PV atata attacks beets beta vulgaris. Pseudomonas syringae PV atrophacians attacks wheat triticum aestivum. Pseudomonas syringae PV disoxyli attacks the Kochka tree Disoxylum spectabile. Pseudomonas syringae PV japonica attacks barley Hildum vulgar. Pseudomonas syringae PV Lapsa attacks wheat triticum aestivum. Pseudomonas syringae PV panisii attacks panicum grass species. Pseudomonas syringae PV papulans attacks crabapple Malus sylvestris species. Pseudomonas syringae PV phaseolicola causes halo blight of bean. Pseudomonas syringae PV pisci attacks Pis pisum sativum. Pseudomonas syringae PV Syringae attacks Syringa, Prunus and Physiolus species. Pseudomonas syringae PV Glycine attacks soybean glycine max, causing bacterial blight of soybean. However, many of the strains for which new species groupings were proposed continue to be referred to in the scientific literature as pathovars of P. syringae, including pathovars tomato, phaseolicola, and maculicola. Pseudomonas savastanoi was once considered a pathovar or subspecies of P. syringae, and in many places continues to be referred to as Pseudomonas syringae PV savastanoi, although as a result of DNA relatedness studies, it has been instated as a new species. It has three host-specific pathovars, Fraxni, Ria, and Oler. Determinants of host specificity equals, it is thought that a combination of the pathogen's effector genes and the plant's resistance genes determine which species a particular pathovar can infect. Plants can develop resistance to a pathovar by recognizing pathogen-associated molecular patterns and launching an immune response. These PAMPs are necessary for the microbe to function and therefore cannot be lost. However, the pathogen may find ways to suppress this immune response, leading to an evolutionary arms race between the pathogen and the host. Pseudomonas syringae is a model system, owing to early availability of genome sequences for Pseudomonas syringae PV tomato strain DC, 3000, P. syringae PV Syringae strain B728A, and P. syringae PV phaseolicola strain 1448A, together with the ability of selected strains to cause disease on well-characterized host plants such as Arabidopsis thaliana, Nicotiana benthamiana, and tomato, P. syringae has come to represent an important model system for experimental characterization of the molecular dynamics of plant pathogen interactions. The P. syringae experimental system has been a source of pioneering evidence for the important role of pathogen gene products in suppressing plant defense. The nomenclature system developed for P. syringae effectors has been adopted by researchers characterizing effector repertoires in other bacteria, and methods used for bioinformatic effector identification have been adapted for other organisms. In addition, researchers working with P. syringae have played an integral role in the Plant-Associated Microbe Gene Ontology Working Group, aimed at developing gene ontology terms that capture biological processes occurring during the interactions between organisms, and using the terms for annotation of gene products. P. syringae PV tomato strain DC 3000 under Arabidopsis thaliana equals, as mentioned above, the genome of P. syringae PV tomato DC 3000 has been sequenced, and approximately 40 hop effectors, pathogenic proteins that attenuate the host cell, have been identified. These 40 effectors are not recognized by A. thaliana, thus making P. syringae PV tomato DC 3000 virulent, that is, P. syringae PV tomato DC 3000 is able to infect A. thaliana, which is susceptible to this pathogen. Many gene-for-gene relationships have been identified using the two model organisms, P. syringae PV tomato strain DC 3000 and Arabidopsis. The gene-for-gene relationship describes the recognition of pathogenic avirulence genes by host resistance genes. P. syringae PV tomato DC 3000 is a useful tool for studying EV-R gene interactions in A. thaliana because it can be transformed with EV genes from other bacterial pathogens, and furthermore, because none of the endogenous hops genes are recognized by A. thaliana, thus any observed EV-R recognition identified using this model can be attributed to recognition of the introduced EV by A. thaliana. The transformation of P. syringae PV tomato DC 3000 with effectors from other pathogens have led to the identification of many ER genes in Arabidopsis to further advance knowledge of plant pathogen interactions. See also, bioprecipitation, ICE-bacteria, Pseudomonas phage I6, NCPPB. References: DiLallo, G., Evangelisti, M., Mancuso, F., Ferranti, P., Marcelletti, S., Taineri, A. and Thaler, M.C. Isolation and partial characterization of bacteriophages infecting Pseudomonas syringae PV actinidiri, causal agent of kiwifruit bacterial canker. Journal of Basic Microbiology. External links: Lava N., Jose, copyright L. Kiel, Christopher. Risano, Owiana. Usseri, David W. Ogeiser, Jose, copyright A. Comparative genomic analysis of two-component regulatory proteins in Pseudomonas syringae. BMC Genomics 8, 397 doi, 10.1186/1471-2164-8-397. PMC 2222644. PMID 17971244.